Welcome back, dear friends. Today we are going to talk about communication between neurons. The electrical changes taking place within a neuron, as described in the previous section, are similar to a light switch being turned on. A stimulus starts the depolarization, but the action potential runs on its own once a threshold has been reached. The question is now, what flips the light switch on? Temporary changes to the cell membrane voltage can result from neuron receiving information from the environment or from the action of one neuron on another. These special types of potentials influence a neuron and determine whether an action potential will occur or not. Many of these transient signals originate at the synapse. Synapses. There are two types of connection between electrically active cells: chemical synapses and electrical synapses. In a chemical synapse, a chemical signal, namely a neurotransmitter, is released from one cell and it affects the other cell. In an electrical synapse, there is a direct connection between the two cells so that ions can pass directly from one cell to the next. If one cell is depolarized, in an electrical synapse, the joint cell also depolarizes because the ions pass between the cells. Chemical synapses involve the transmission of chemical information from one cell to the next. This section will concentrate on the chemical type of synapse. All synapses have common characteristics, which can be summarized in this list. First, presynaptic elements. Second, neurotransmitter packaged in vesicles. Next, synaptic cleft between presynaptic element and postsynaptic. Okay, synaptic cleft. Receptor proteins, postsynaptic element, and neurotransmitter elimination or reuptake. For the neuromuscle junction, these characteristics are as follows. The presynaptic element is the motor neuron's axon terminals. The neurotransmitter is generally acetylcholine. The synaptic cleft is the space between the cells where the neurotransmitter diffuses. The receptor protein is the nicotinic acetylcholine receptor, and the postsynaptic element in the sarcolemma of the muscle cell, and the neurotransmitter is eliminated by acetylcholinesterase. Other synapses are similar to this, and the specifics are different, but they all contain the same characteristics: presynaptic, cleft, postsynaptic, and neurotransmitters that binds with receptors of postsynaptic membrane. Neurotransmitter releases. Neurotransmitter release. When an action potential reaches the axon terminals, voltage-gated calcium channels in the membrane of the synaptic end bulb open. The concentration of calcium increases inside the end bulb, and the calcium ion associates with proteins in the outer surface of neurotransmitter vesicles. The calcium facilitates the merging of the vesicle with the presynaptic membrane so that the neurotransmitter is released through exocytosis into the small gap between the cells, known as the synaptic cleft. Once in the synaptic cleft, the neurotransmitter diffuses the short distance to the postsynaptic membrane and can interact with neurotransmitter receptors. Receptors are specific for the neurotransmitter, and the two fit together like a key and lock. One neurotransmitter. Receptors are specific for the neurotransmitter, and the two fit together like a key and lock. One neurotransmitter binds to its receptor and will not bind to receptors for other neurotransmitters, making the binding a specific chemical event. The synapse is a connection between a neuron and its target cells, which is not necessarily a neuron. The synapse is a connection between a neuron and its target cell, which is not necessarily a neuron. The presynaptic element is the synaptic end bulb of the axon, where calcium enters the bulb to cause vesicle fusion and neurotransmitter release. The neurotransmitter diffuses across the synaptic cleft to bind to its receptor. The neurotransmitter is cleared from the synapse either by enzymatic degradation, neuronal reuptake, or glial reuptake. Neurotransmitter systems. There are several systems of neurotransmitters that are found at various synapses in the nervous system. These groups refer to the chemical that are the transmitters, and within the groups are specific systems. The first group, which is a neurotransmitter system of its own, is the cholinergic system. It's the system based on acetylcholine. This includes the neuromuscle junction as an example of a cholinergic synapse, but cholinergic synapses are found in other parts of the nervous system. They are in the autonomic nervous system as well as distributed throughout the brain. Another group of neurotransmitters are amino acids. This includes glutamate, gamma-aminobutyric acid, a derivative of glutamate, and glycine. Another class of neurotransmitter is the biogenic amine, a group of neurotransmitters that are enzymatically made from amino acids: serotonin, dopamine, norepinephrine, and epinephrine, or adrenaline and noradrenaline. Next group is a neuropeptide. Is a neurotransmitter molecule made up of chains of amino acids connected by peptide bonds. Some neuropeptides are quite short, such as methencaphaline, which is 5 amino acids long. Others are long, such as beta-endorphin. Others are long, such as beta-endorphin, which is 31 amino acids long. That's it. See you later. Bye-bye.